Today, we're going to talk about crossing the membrane. Now remember guys, we've already talked about diffusion and osmosis. We've learned that the concentration gradient drives the movement of water molecules across that semi-permeable membrane, but not all molecules have the same ability. And in fact, some of them need some help. Proteins are the helpers. There are embedded proteins that are tunnels made by those proteins, and then there are peripheral proteins and carrier proteins that help carry molecules across the membrane by changing the shape of the molecule and allowing it to pass through those phospholipids. In facilitated diffusion, the use of carrier proteins is used, and diffusion then occurs. This doesn't require any energy, so it is called passive transport. Glucose enters and leaves the blood this way. In active transport, we need energy. Think of active as needing energy. That's energy being used to transport the molecules across the membrane. And most often, this occurs when the electronic charge is opposite of what it needs to be to move freely across. It involves the use of carrier proteins that act as pumps using energy to pull things across. The most important and famous of those pumps is the sodium-potassium pump that fuels nerves and nerve impulses, muscle contractions, and the absorption of nutrients. In plants, active transport allows the roots to pull in nutrients from the soil, even though those nutrients could be very densely concentrated. And so the concentration gradient would be for them to move outside of the cell, and so they need energy to pull them in. Think of yourself stuffing yourself into a crowded elevator. It requires energy. This is the type of energy that is used. Now, when we talk about bulk transport, this is moving large quantities of molecules across the membrane by putting them in membrane-bound bundles or pouches and then moving them across. The molecules are too large to cross the cell membrane, and so the membrane itself literally forms a sac around them and pinches in. There are two kinds. Endocytosis is when the cell membrane folds into a pouch and brings molecules into the cell, either by pinocytosis, which is when the cell membrane encloses fluid and brings it into the cell, or phagocytosis, and that's when it brings food particles into the cell. So in endocytosis, either liquids or solid particles are moved in via pinocytosis or phagocytosis. And I always think of phago with the PH sound and food. So I remember that phago is food and pino is fluid. And finally, exocytosis. This is used exceptionally by the Golgi apparatus. The Golgi apparatus packages materials into little packets and then shoves them out of the cell. So they're removed from the cell in the process called exocytosis.